We would like to welcome you to the Sanguine Speaker Series webinar, Big Data in Clinical Research, Sharing, Dissemination, and Repurposing. Today's webinar is presented by Sanchita Bhattacharya, the Informatics Project Lead at the Bakar Computational Health Sciences Institute at the UCSF School of Medicine. I will now hand it over to Sanchita.
Thank you for the nice introduction. Good morning from San Francisco. Good afternoon on the other side of the coast. Thanks for inviting me, and I'm really excited to present our work to this audience. And today I'm going to talk about the big data in clinical research, sharing, dissemination, and repurposing of open access clinical and immunological data. So clinical trials generate vast amount of data. A large portion is never published or made available to other researchers. On the other side, for last few years, there's a lot of initiatives going on for data sharing, which could advance the scientific discovery and improve the clinical care by maximizing the knowledge which are gained from the data collected in the trials, stimulating new ideas for research and avoiding unnecessarily duplicative trials. So in 2015, IOM, Institute of Medicine, part of the US National Academy of Sciences, released a report which provided a guiding principles and a practical framework for the responsible sharing of the clinical trial data. The chart outlined here shows the major stages of the clinical trial life cycle and what data should be shared, when to share, specific data packages in common scenarios in order to help amplify the scientific knowledge while minimizing the risk. As you can see here, that different types of data were in various stages of the life cycle. The data is getting collected like metadata. There's the data about the data, like protocol, statistical analysis plan, analytical code, and the data, the metadata collected on the individual participant data, that's called the subject level or a row level data. That's the data that are collected from the participants. Primarily, I'm referring to the raw data, which is then further cleaned and abstracted, coded, and transcribed to become an analyzable data. And then that's called mostly the summarized data. And then, once all the data is analyzed and ready for publication or submitting it to the regulatory agencies, there are post-publication data packages, full data package. And so you can see here the breadth of data that is getting collected during the clinical trial life cycle. On the other side, and then, as I mentioned earlier, that industry and regulatory forces are driving these initiatives to publicly share the patient level data from clinical trials. And there are a lot of electronic portals getting built to share the data. And as it's been, that's the platform for, kind of generating better evidence, facilitate additional findings, confirm the published results, and ultimately improve the public health. On the other side, there's a lot of data. When I say individual data, I'm also referring to the mechanistic data. As you know, that immune system is one of the very complex and dynamic biological system, which is comprises of diverse cell types, cytokines, and immune components with varying functional states. So there are lots of data getting collected at the population level and a single cell level on the proteomics, transcriptomics, and genomics and epigenomics, and there are a lot of high throughput technologies collecting the data. On the proteomic side, the flow cytometry, and then lately our single cell level data getting collected on the CyTOF platform, mass spectrometry, and then microarrays. Tons of this data was getting collected in the last decade on microarrays, and now moving to RNA-seq and single-cell RNA-seq, DNA-seq, ChIP-seq, and you name it. And so we are definitely in data deluge here with lots of data from high-throughput data getting collected, but how we transform this into knowledge. So there are over 2,000 open access data repositories. Typically, the generalist repositories like Figshare, you might have heard these names, Figshare, Dryad, Zenodo, Mendeley. The publication data is getting uploaded to these repositories to share the data or the share the data from the table generated in the publication. The underlying data is getting submitted to these generalist repositories, but there are also open and domain specific repositories like Geo and SRA and a lot of other databases. And as you can see here, the data access, there are a lot of levels, like some are open, some are restricted, embargoed, or there are data access restrictions, you need to register, or there's a fee required, or there's an institutional membership. So some of these have some requirements and also the data upload side, not only just data access, but data upload. Again, registration, or you need to be a part of a consortium, and there's a database around that consortium where they're submitting the data. And then I already mentioned about the generalist and the domain specific like GTAC, TCGA, and many others. Today, I'm going to highlight one such repository, Import, that allows open access to clinical and immunological data sets. And I'm the scientific program director for Import, so you're going to be hearing more about the initiatives we have taken to repurpose the data that is submitted to the Import database. But next few slides, I'm going to talk a little bit about the database. So Import Data Portal was developed to collect and share research and clinical trials data from the National Institute of Allergy and Infectious Disease Division of Allergy and Transplantation Funded Research about over a decade ago. And so to promote the FAIR principles of findability, accessibility, interoperability, and reusability of the data. And so import ecosystem comprises of like private data. That's a private workspace where the data providers, data submitters, they upload the data. And once they are ready to share the data is embargoed till we hear from the investigators that they are ready to publish their paper and release the data to the research community. Then it goes to the shared data space. And then we also have resources and data analytical tools that's under data analysis. We have workflow, and as we go, I'm going to explain a little of these analysis tools that we have and the resources that we have built over the years. Regarding how to access import in Fairly, it's very easy. If you have an email of your choice, you just register, just provide your name with an email address, and you will be given access to these data sets. There's no proposal, which we have seen in other databases. You need to write a proposal, and there's a committee to review the proposals. But on the other hand, for import, it's just we need a email address, valid email address, name, and you are good to go. So who is distributing the data? That's quite often I get asked that, where are you getting all this data from? So Import redistributes the data from major NIAID funded programs. And as you can see here, the list, we have a long list of different NIAID programs. Those who are submitting the data, we have over 30 plus programs submitting clinical and basic immunology research data through this portal. And lately, we also have some COVID-19 data coming to import. I have highlighted few on the left-hand side with the logos like Human Immunology Project Consortium. They're a big data contributor. Through this program, a well-characterized human cohorts are studied using a variety of modern analytical tools like transcriptional, cytokine, proteomic assays, multi-parameter phenotyping of the leukocyte subsets, and assessment of the functional status of leukocytes. We also work with Bill and Melinda Gates Foundation. So we have TB data coming from BMGF Foundation, March of Dimes for the preterm birth research data. And then we also have data from AMP, that's the Accelerating Medicines Partnership in Rheumatoid Arthritis and Lupus. It's a joint collaboration between NIH, NIAMS, that's the National Institute of Arthritis and Musculoskeletal and Skin Diseases, industry and academia coming together, collecting high throughput multi-omics data, such as CyTOF or bulk or single cell RNA-seq, and many more from those arthritis and lupus patients. We also have some cancer data, cancer immunotherapy data that's coming from the Oncology Models Forum, which collects the data from various cancer models, genetically engineered, transplantation induced, and spontaneous mammalian models. So as you can see, the breadth of data, and there are many more for the that we are getting a lot of transplant data as well, and they're all getting submitted and shared through import. So the focus areas and data types in import, just to give you a flavor of the type of studies. As you can see here from the pie chart that we are heavily on the vaccine response. So we have about 143 studies on vaccine response, immune response, infection response, transplantation, autoimmune diseases, allergy, preterm birth. And regarding the diseases, we have roughly about 112. Most of them are related to infectious disease. We have influenza, aging, transplantation, and then we also have a lot of pregnancy data, smallpox, lupus, dengue. Um, and in terms of looking at the experiments, the type of, we support multimodal data, not only just one data type, but then we support flow cytometry, we are one of the largest repository on for flow cytometry, and transcript profiling, ELISA, Luminex, all these immune measurement techniques. So we support RNA-seq, CyTOF, we have lots of CyTOF data, and then HLA typing data, SNP microarrays, and so on. And then here is the distribution of the human subjects. As you know, most of the database is a little bit biased on the white population. And then we have other races and ethnicities as well. Immunology research, we have about 305 studies, and we have, there are quarterly release of new studies. So every quarter we have somewhere from, it depends, but somewhere between 10 to 20 studies get released, and sometimes more. So the data summary that I'm showing you today is from September 2020, and there's very soon we'll be coming out with the next data release. And we have about 140 clinical trials. A little bit about the data model. So import database is a relational database. So the data is stored in tables, and import database is very study centric. So, we will start with a study, we describe a study, and then within a study, let me just give you an example that, a study has subjects, and then subjects also belongs to a cohort arm or a cohort, which means treatment arm versus control arm. And from the subjects, you can have the biosamples, and biosamples could be like whole blood, for example. And then from whole blood, you isolate the serum or plasma. So that is considered to be an experimental sample. You run the experiments on these experimental samples. The protocols from the experiments are also curated in the database. And so from experimental samples, there are different types of experiments getting carried out, from ELISA to flow cytometry and many other immune measurements. One of the other things that I would like to mention that, when you're looking at data harmonization, the data standardization is very important. So data standardization and use of ontologies. So on the extreme right, you could see that we use a lot of these different ontologies to standardize the data, because this data is coming from different providers, and it's diverse, right? Coming, and we do have, and I'll show you how we collect the data. But then these ontologies helps you to standardize the data. So the data ingestion is in the form of like templates. These are the data templates, exactly the tables that I showed you before when we give the data providers these data templates, and which helps them to populate these templates. And then once they populate the templates with all this information, some are required information, some are optional, so mandatory versus optional columns. As you can see, the ones in the gray is optionals, but they are required columns, and these are the data templates. Through these data templates, they submit the data to import. In the interest of time, just wanted to give you an idea about how the data gets ingested into import. So once the data is in the import, as I said, from different programs, now I would like to show you, this is one of my favorite slides that I would like to show the breadth of the data, as well as in a unified way, how you can leverage all these data sets. So this is an example of the influenza vaccination cohorts in import database coming from different sources. Yet you could see here that the bubble chart illustrates the variety of these open access flu vaccination associated studies in import, and the big blue circle here consists of the healthy vaccinated individuals, and the number represents the number of study participants. So the data sets can range from a niche data set to fully integrated data generated on a variety of platforms. And behind you have like flow cytometry, microarray sequencing, ELISA on thousands of patient samples. But this clearly demonstrates the accessibility of large volume of data sets within an area of interest in one central location. You can see healthy, you have a lupus cohort around a common theme of all of them are like, flu vaccinated cohort with, either a comorbidity or other features like, there's some aging population, twins, if you had doing some genetic studies on vaccinated individuals, but these are twins, so you could, study the genetic as well as the environmental differences. So, or a pregnancy cohort, transplant data. So this clearly, with the increasing awareness of the importance of the sharing research data and research findings, there's an additional need to showcase how to best leverage these shared data sets across research domains. So through case studies, our group has successfully demonstrated the major efforts in meta-analysis of these open immunological data to build knowledge base, gain insight into discoveries, and generate data-driven hypotheses, those which were not originally formulated in the studies. And here, this is a cartoon where you could see that, the investigator, the primary investigators, they collect the data, it goes to the repositories, and then bioinformaticians like us download that data. We come out with new novel hypotheses. Again, these are results into secondary publications. We do validations, and then, we publish papers, goes back to the database. And there is a paradigm shift. Most of the time you start the experimentalist or basic immunologists, they start with a priori hypothesis, they run the experiment. We are more coming from the looking at the big data, data-driven hypothesis generation, and then, bring out a model and find out some novel discoveries or novel findings. So now I'm going to give you, next few slides is going to be all about the use cases, how we have leveraged the data. So the first one is a rave analysis. So this paper came out back from our group in 2015, where this clinical trial data was submitted to import. And then that's called the RAVE trial. It's an immune tolerance ITN trial for rituximab, the role of rituximab in ANCA-associated vasculitis. So I'll just go over a little bit on about the trial. The paper was published. The first paper published on this trial was by Stone et al. In New England of Journal of Medicine in 2010. And the trial is a randomized trial, double-blinded, active controlled phase 2/3 trial, where they were comparing rituximab to cyclophosphamide for the induction of remission. As you can see on your left, that there were two groups, rituximab and cyclophosphamide, and they rituximab plus glucocorticoid given to these subjects, 2 arms, 30 visits. They were followed up to 18 months, and there's a study data. Data was collected from both the treatment groups. There was a remission induction phase and the remission maintenance phase. And the types of data that was collected on the clinical side was the assessment, lab tests, adverse events, concomitant medications. And then on the mechanistic side, they ran the flow cytometry panels, ELISA, and the gene expression data. As you can see, lots of data. And all this data by phase went into import. So this trial had about 197 participants. It was a multi-center trial. So what was observed and published by Stone et al. Was that 63 of the 99 patients in the rituximab group, which is roughly about 64%, reached the primary endpoint, that is the complete remission, as compared to 52 out of 98, which is like 53% on the cyclophosphamide group. So this is the data we got, and that's what we learned from reading the paper. Once the data went into import, we downloaded the data, and I would like to walk you through the process that as a bioinformatics scientist, or data mining, how we download this data. So once we download the data and reading the paper, the question that came to our mind is that 35% of the patients treated with rituximab and 47 treated with cyclophosphamide failed to achieve remission. So retrospectively, do any measured factors predict the response to therapy? Can we find out from this raw data submitted that what might predict response to therapy and why these patients didn't achieve remission? So the process was that since the data was available, we looked at the clinical data from the first 6 months, and then the mechanistic data. We downloaded the flow cytometry data. We also downloaded the anchor titers, which were available from this data. And then there were flow cytometry data. We had 15 pounds, 36,000 files. So we used an automated gating approach to do a high throughput analysis of this flow cytometry data. And the very first thing that we did is that since the paper was published, we wanted to validate that all the data that we downloaded kind of match with what is reported in the paper. And so in the paper, they did a manual gating, and we kind of reproduced the same results using the automated gating of the flow cytometry data. And as you can see here, the graph on the left as well on the right, that they match very well. So that gave us a lot of comfort in terms of like, yeah, we can proceed further with this data because we were able to reproduce the results. And then we went further and we asked the question, because that was not asked by the primary investigators, how about looking at the data at the baseline? No. So before treatment, we would like to see, and since we were fortunate enough to have that baseline data, we wanted to see from the flow cytometry data, is there any difference? So what you see here in the plot, the Wallin plots, that we have two groups, rituximab treatment group and cyclophosphamide treatment group. At the baseline, we looked at the granularity index. So basically, we were focusing on the granular sites and we saw some differences. And what we saw here, as you can see here, that within the granular sites, there are subsets. Some have low, we call it low granular sites, meaning hypogranulated, and high means hypergranulated subsets. And so as you can see here, that there is a gradient. So the ones which are in light pink or to dark green, light pinks are only hypogranulated, and those which are in the dark green are hypergranulated. So now if you look at the rituximab group and the cyclophosphamide group, In the rituximab group, if you look at the x-axis in the success, those who achieve complete remission have a little bit higher, or it's a low granulocyte, means hypogranulated, versus those who were in the cyclophosphamide group, they were more on the hypergranulated side. And that's what we show in the Wallin plots, where you can see here that those who achieve remission in the rituximab treatment group, they had kind of if you see the granularity index, you would see that they had a little bit higher compared to the cyclophosphamide group, where they had a lower granularity index. So kind of hyper and hypo. We just, for the sake of simplicity, we just termed it as a granularity index. And so here with these 187 subjects, then we were able to partition them. And as you can see here, GI index, and look at the rituximab and cyclophosphamide groups. And basically, this is kind of like more of a personalized treatment option based on the cellular profiling and substudying the groups. And we can predict now, based on those who are hypergranulated at the baseline, you could see then you take this route and who is going to achieve remission, and those who are less, how you go on this side, on the left side of the arm, and who is going to have a complete remission. So basically, we give a kind of a more treatment option, personalized treatment option. So the current method from the first paper was like, it's a non-profile therapy. It's like one size fits all. But then we proposed a method based on the granularity index and how you could have a profile therapy. So this is our first paper about reanalyzing the clinical trial data and come out with a like novel way of treating the patients. So this is so that in the follow-up trials, the clinical trialists, those who design the clinical trials can take this into consideration for the future trials. Now I'll move on, and so based on this success, then we embarked on another project, and this paper came out in Cell Reports in 2018. We built from this input data a 10,000 immunome project. It's a building a resource for human immunology. So to date, however, so the motivation for this project came from resources such as 1,000 Genomes Project, Wellcome Trust Case Control Consortiums, and others who have uniquely enabled the understanding of global variation in human genome in health and disease. To date, however, human immunology has no such resource. And given the recent growth in open immunology data, and as you can see that lots of data getting collected to import, this was an attempt to synthetically construct the reference immunome by integrating the individual level data from publicly available immunology studies within import. And as you know, that moreover, the immune assays have not been reproducibly characterized for a significant, large and diverse healthy human cohort. So what I mean here is the baseline data. So what we built was a large, diverse, clean reference data set, and then interactive data visualization, custom control cohorts, and standardized data download. That's what we have provided in the 10,000 immunome project. Let me walk you through this process. So we did a manual curation of the studies, the arms, control or treatment arm, and plan visits to filter for normal human subjects. So we just subset the data within imports coming from different studies. We just looked at the control arm, so-called normal healthy individuals. So there were about 242 studies when we started this project with 44,000 subjects, 290,000 plus samples. So we looked at only control arms with no manipulation, and then we filtered for only normal controls. We went over the import study design, read the inclusion-exclusion criteria. We even contacted the authors, those who submitted the data to import, if we had any, for the sake of clarity, and then we also read the protocols. So after going through all this rigorous filtering process, we were left with 85 studies. And as you can see here on the right, each study in import has the study design and types of data getting captured. So we manually read all of these study. And then from these 85 studies, we had, so the type of data those were collected are like side-off and flow cytometry data, the more mainly the cytokine data, and then other data types, gene expression data. So we standardized all of data. We had a standardized pipeline for data cleaning and harmonization. So basically, automated grading for flow cytometry. We also had to standardize the cell subset names, and then validate it against gold standard hand-gated populations. So a lot of these data standardization and harmonization went behind all this data curation. The table on the left is, the number of samples, number of subjects. We had 10,000 plus subjects and type of measurements for, and the types of data, right? These are the ones where you can look at ELISA, virus titers, clinical lab tests. We also collect lab tests. So we had CBC counts, metabolic panels, lipid profile, cytometry, and so on. Just to give you an idea about, we have a huge, like a pretty wide age span, somewhere from newborns to older cohorts, and then also in terms of ethnic groups, and we have a good distribution, but as you mentioned before, that each of these databases is a little bit biased about the white population, and then followed by Asians and others. So basically, after the standardized data for data dissemination, whatever we learned, we curated. We have a website called 10,000immunomes.org, where you could go to this website, and based on your data of interest, this is all built on the R Shiny platform, and immunoassays or transcriptomics, proteomics, depending on your interest. You can look at the data, and you could do all this in real time. This is an example of the interactive property. This is a screenshot of the web-based 10K immunome resource. So the graph updates in real time, the subject counter updates, and then if you can select or unselect what you are not interested in, or plot by ethnicity, by study as well, and age and sex. And so that's kind of gives you an idea. And as well, you could download. So you can download the image, the data is plotted here, and all of the data. So this is all, as I said, are standard. So now, what you could do with this. So when we published the paper, we wanted to show as a use case that you could build like cell cytokine networks from these baseline data. Or there was one interesting study in import where the primary investigators were looking at the variation in serum cytokines over the course of pregnancy. So they were looking at the immune perturbations during pregnancy. And so this study had been published previously. However, what we observed that the study design did not incorporate a pre-pregnancy control. As you can see here, that they had the data from first trimester, second trimester, and then 6 months postpartum. So they had all these data, but they didn't have any data before pregnancy. So pre-pregnancy data was missing. So using this human, baseline data from 10,000 immunomes, we were able to show, as you can see here, that's been highlighted in the red box, that we were able to plot the data to see if someone is interested to know what was the pre-pregnancy, the control data, though these are from different women, but still you can control in the same plot to see the differences and the p-values of what are the changes happening over the pregnancy period. So this is one of these use cases that even if you don't have the control data, you can get the information, you can download the data from resources like 10,000 immunomes, which has the baseline data, and can plot it with your own data. So this is another project from Import, where this time we leverage the data on living donors. Mostly you hear a lot about the recipient data from transplant recipient, but this is like the data on for living donors. In import, we have about 27 clinical trials on organ transplantations. So we curated 20 of them, and there were some selection criteria. We didn't include the deceased donors. We were only looking at the living donors. And then only we took the data with 95% missing those over more complete data, and eliminated where we didn't have enough data. So with those selection criteria, we were left with about 11,000 subjects, and as you can see that these data were coming from different studies. We compiled the data. The data was standardized, and the data that we collected were demographics. The data that we curated were demographics data, pre-transplant, the data that was available, and then interoperative post-transplant data, and also the relationship of these donors with the recipient. So the question was that what can you do with this data? So we then thought about how about we can do a trajectory analysis. So this is the trajectory analysis that that I'm showing you here, as like, these are all the living donors, and they were followed, and we looked at the data, and some of them were followed somewhere between 2 years after they donate, and this is kidney transplant data that I'm showing you here. After they donated the kidney, they were followed somewhere from 2 years to 40 years, right? So what we have plotted, and this is a trajectory analysis of these living donors, and it's color coded based on if they had any surgical complications, that's in red, and then the ones, those were non-surgical in light blue. And first 2 years, you mostly see a lot of surgical complications. As the year goes by, we saw that in living donors, they come up with other secondary complications, which are mostly non-surgical, and some of them are going back to the waiting list. That is unfortunate, but we just are reporting the data that was submitted and what was reported. So here the data is all based on what were reported in the questionnaire, and somewhere in the long-term maintenance dialysis, and so on. So this kind of gives you an idea about the trajectory or the path once what the living donor is going through in the medical conditions. And this resource is also available in Import if you are interested. Um, and then the next project that I'm going to talk about, let me see how are we doing with the time. Okay, this is about a robust and interpretable end-to-end deep learning model for cytometry data. And as I mentioned before, that we are one of the largest repository for flow cytometry data. So we started using the deep learning to understand some of the cytometry data in Import. So we built a convolutional neural network data from cytometry data. So here's this data that I'm talking about is mostly on the site of data, and basically the cell surface markers and the cell. This is a matrix. We have different convolution layers. We pull the layers, and also we relate the cytometry data with the demographics. Non-cytometry data could be any clinical condition or demographics data. So we would like to see the influence of the cell subset populations based on the clinical phenotypes. And so data set here in this case contains the site of, and we also have the serological data from cytomegalovirus serological data from 472 healthy individuals across 9 studies. We had a training set, and these are coming from different studies. training set, validation test, and then testing set. And so we were looking at CMV, which is a latent infections found in normal healthy individuals. So there are two populations here: those who are CMV positive, CMV negative. These were um, and this was based on the serological data that they were kind of divided into two groups, and then training, validation, testing. So the goal was to diagnose the latent cytomegalovirus in healthy individuals using the deep learning model. So what we did is that, if you look at this matrix, we had the cells on the rows are cells and surface markers are in the columns. So this is the original data. And then we did some upsampling of this data. And then that's called the modified data. And so this is the workflow for interpreting the full deep CNN model. So once we upsample the data, we calculate the changes in the model output. So original data, modified data, we look at the delta Y. That's the difference between the original and after the upsampling. And so we did this based on, this is a decision tree that we use to interpret the output from the CNN model. And the CNN model actually identifies the association between the immune cell subsets and the CMV infection. So here is the decision tree, which identifies the cells that lead to the largest changes in the model output that I just described here, the delta Y, when it was upsampled, and each node represents a cell subset, and the rules by which this population split are indicated inside the nodes, and the values in each node represent the percent of the subset in the total population of the average change of the model output. And what you see here, the red box highlights the node with the highest mean of the delta Y. And so that's how this is the permutation-based method to interpret the deep CNN model. And using this approach, we were able to identify a cell phenotype which is associated with the CMV infection. And as you can see here, the graph, the bar plot shows the box plot shows you the difference between the CMV negative and CMV positive subjects, and we consistently observe that there is this phenotype CD3 positive, CD8 positive, CD8 27 negative, CD8 94% positive cells. That cell phenotype is associated with the CMV latent infections in positive individuals. So this is all we derived using the CNN model. So basically, just to give you an idea that using the deep learning, you can identify these cell phenotypes. And we have also provided the deep learning model tutorial in our paper, which was published in 2020 in PNAS from my group, which helps you to kind of, go through this process, this pipeline. And so basically we use the keras and TensorFlow to build or make this tailored deep learning model. In the interest of time, if you have any, we can always talk offline if you want to go into details. Let me move on and show you another project. This is an unpublished work, but we are now leveraging real-world flow cytometry data from electronic health records. And in the last couple of years, the research community is witnessing a wide adoption of electronic health record systems, which generates the big real-world data. And so this opens up new avenues to conduct clinical research. So what you see here is that we have developed a tool for comparing. I have shown you before the 10,000 immunomes, which was a baseline healthy individual data. So we were comparing the 10,000 immunomes cohort to the data sets from EHR, and this is from UCSF electronic health records. We had over 4 million patients, and out of this, we were looking at and leveraging the clinical flow cytometry data. So we were very excited to see that that much of data was available on the clinical flow cytometry data sitting in a database where we had the lymphocyte subsets like CD8, CD4, CD56, and CD19 cell pop subsets from over 100,000 like peripheral blood samples in 41,000 patients. So this is all coming from the patient's medical history, and we had demographics data, diagnostic codes for all these lab tests, and so we were comparing with the baseline healthy individuals to the ones where mostly when, since it's a EHR, we have mostly patients with certain conditions. And so if you see on the right, we had data from immunodeficiency, primarily a lot from the HIV group, transplantation, infections, metabolic disorders, autoimmune disease, blood disorders. This paper, we are about to submit this manuscript, but I can share some of the preliminary results. Here is the CIDR app. It's not yet public, but it's a simple tool which will enable the researchers to easily study the real-world population in EHR, and which would serve as a model for other types of data. So what you are seeing here is the CD4 positive cells in patients with stem cell transplant status, and then we are comparing it to the healthy individuals from 10,000 immunomes. The ones you see here in a bin plot. On the left is 10,000 immunomes baseline data, and EHR data is on the right. And then we did run a regression analysis elucidating the different variables associated with the perturbations in cell count. And you can see then when these are the regression coefficient. And in the interest of time, I'll just show you some of the additional analysis we did from this data set. We were looking at the CMV disease and the comparing the PBMCs, the cell counts in patients with stem cell transplant. This is all stem transplant, but then along with stem cell transplant, they also have CMV disease. 0 with no disease and the 1 with CMV disease. And then we looked at the effect size of the CMV comorbidity and then other autoimmune diseases, and looking at the differences in the CD8, CD4, CD19, CD56, all these different cell subsets. So just to give you a flavor of clinical flow cytometry data from the real world data, how you can, and this can transform into knowledge of data mining for future studies. So I just want to give you some of the pointers that if you are interested in the resources, go to the import website, which is open for all. And if you go to the resource tab, whatever we learn, whatever the projects that we try to accomplish, and then publish it, and also we disseminate the information to the research community. So you can check out the import resources. We also have tutorials, those who are bench immunologists or who are willing to learn more about how to use the APIs. Import has APIs. If you want to retrieve the data using the programming interface, you could use the import APIs, or you would like to learn basics of programming, we have a lot of tutorials. And also, if you have some interesting data, you can always contact us. I just want to leave you with some thoughts on the opportunities and challenges in democratizing the clinical research data sets. Here is the time, and there are a lot of data sets sitting in silos. There's a need. This is a call for the research community. Let's come together. Let's democratize the clinical research data sets, which are either like, data sets sitting in different repositories, and then there are different tools, but how to improve on the accessibility, discoverability, and interoperability. I think I'll end here with the take-home message that I wanted to give you a flavor of holistic approach to analyzing clinical research data. Open access immunological studies are a valuable resource to evaluate new in silico hypotheses, gaining novel insights. 10,000 Immunome Projects is one of them. It's a framework for growing a diverse human immunology reference, which allows us to learn from the features and candidates that we already know, and it enables us to explore new factors to be discovered. Deep convolutional neural network model I've shown you has accurately diagnosed the helps you to diagnose the latent CMV infection in healthy individuals. There's a vast landscape of clinical flow cytometry data and disease that could be explored through this app to generate new hypotheses. And I would like to end with that embrace open access data sets. There's a lot there and which can give you novel insights to the data. So please embrace the open access data set, and hopefully I was able to convince you. And I will list but not last the acknowledgements. Dr. Atul Biud, who is the PI for import, and he's also director of the Baker Computational Health Sciences. I would like to thank the data providers. Without that, we couldn't have done this. And then we work with Northrop Grumman Health Solutions. Those who are the data curators, they maintain the database. This is a contract through NIAID, National Institute of Allergy and Infectious Diseases. So we are always in touch with NIAID program managers for all these projects that we launch. If you have any questions, please contact the help desk at Import. And if you're interested for more about big data, learning about big data in immunology, I'm offering, and every year I offer a workshop at the Focus workshop, and it's coming up this year on June 28th. So if you want to attend a hands-on session, how to retrieve the data sets from import or mining import data, feel free. And with that, I would like to thank and happy to take questions.
So we have several questions that have come in. Start with, can you discuss the COVID-19 database if possible, the strengths, the weaknesses of the public portal data that is available?
Sure. So we have about 5 data sets from COVID-19, and we are still building. The strength is, I would say, that row level data, the subject level data that we are curating. So the data that's going to import, and I have shown you that there is so much of wealth of information available when you are looking at the raw data and subject level data, where you could do a lot of these baseline analysis and personalized data, right? So that I would say a strength, and it's raw data. We also have some summarized data, but raw data helps you if you have your own questions and you would like to start from scratch. Well, in terms of weakness, well, that's more, I wouldn't say a weakness, but it's more about it takes time to curate such data, because, as you're getting from the data submitters, data standardization takes some time, data harmonization. So I would say a little bit time consuming. So we are not able to right away share the data. It takes a little bit of time when you are trying to integrate the data coming from different sources. So I would say I would put a lot of emphasis that, it takes, and I would like to give the credit to the data curators, those who are trying to standardize the data. And then I am more on the receiving end, where I was showing you all these projects where we were able to integrate the data. But data standardization and data harmonization is the key to all these data mining projects.
The next question is, how can researchers be incentivized to collect data in a manner that supports repurposing down the road?
Right, so we are getting a lot of these questions, and yeah, definitely, as you have seen my acknowledgement slide, that I always acknowledge the data providers, because it's so important that if they don't share the data. I mean, all this, as I said, it will sit in silos somewhere, but it's one way is that we are talking about, like, these are NIH funded programs. So, NIH incentivized by, if someone is sharing the data, maybe their proposal somehow will be expedited, or, some level of, when they are scored, data sharing will be one of those which will be taking into consideration, and incentivize also by, when we are writing the papers, right, the publications, we always acknowledge the data providers, because they are the ones, they were at the front line, they came up with the papers, they generated the data, and then we reuse the data. So we should always be thankful to them, and also sometimes offer the authorship. So we also have included in some of our papers the primary investigators as our co-authors, if that answers the questions.
The next question is, what portion of the data in these libraries originate from industry-sponsored clinical trials? Thoughts on incentivizing sharing of data by pharmaceutical companies from such trials, at least from the placebo arms and discontinued programs.
Right. So, we are not directly taking the data from the pharma, but these are primarily NIID funded sponsored trials. But some of them I have seen are partnered with the industry folks. And so you will see some of the trials which are partnered between the academia and industry. Those trials go to import. But if you are looking for solely industry-sponsored trials, there are other websites like Vivli. Vivli is sharing the clinical trials from the pharma. So I would say these are mostly the trials in import are industry and academia collaborations.
And for the last question, how does big data affect clinical trials, sample management, and data reconciliation?
I don't think I am capable of answering that question, because I am more on the receiving end, looking at all the data that is already being curated. That is more a management question. And we work with CROs. So import work with the CROs, and so RO is one of them, and we have many other CROs. So we get the data directly from them, and so it's more about when the clinical trial, the sponsors agrees, then it goes to RO, and then we just get that from the RO. So I think I won't be able to answer that question because it's more a management decides on the samples, the sponsors basically.
Great, thank you. That is the last question.
Thank you, Sanchita. And thank you for joining Sanguine for our S3 webinar on Big Data in Clinical Research, Sharing, Dissemination, and Repurposing. For a list of upcoming webinars or to request patient samples, visit sanguinebio.com. Thank you again, and enjoy the rest of your day.